This video is Western 2, and it will focus on steps 1 and 2 of the Western blot. This most specifically ties to these two SLOs. You should definitely watch the first Western to make sure that you cover all the information to answer the SLOs. So step 1 is the sample preparation, which is making the lysate, and basically this includes a few key ingredients. So for sample preparation, we use a detergent, and if we wanted to be able to look at membrane proteins, specifically those that are embedded in the membrane, then we would definitely need to use a strong detergent, which would dissolve or disrupt the cell membrane. And that solubilizes the membrane proteins such that now they would be in the slurry instead of in the phospholipid bilayer. This also means that all those proteins would be individualized, so not in any large protein complexes, and so we could see those individual proteins on a gel. This is what a detergent monomer might look like, so a hydrophilic head with the single hydrophobic tail. And then in the schematic you can see a membrane protein here, and you can see in darker green the nonpolar amino acids, and in green the polar amino acids. And it's sitting in a lipid bilayer. If we've got our detergent monomers over here, which again, see that it's a single circle with a single line, they can form these little micelles or micelles, which are all oriented towards each other, and those will spontaneously form in a aqueous solution as they are likely to form stable interactions with each other and be excluded from water. And in the presence of things, in addition to the detergent, to help break up those membrane proteins, we could use things like a vortexer and heat, something like 100 degrees Celsius. Then we could start to see that the membrane would break down and the detergent monomers would start to cluster around the nonpolar amino acids. To get that ready to load onto a page and do the Western blot, then we would centrifuge down that slurry, and then we would get a pellet of insoluble materials like lipids and whatever sort of other crud. But the liquid portion, or the supernatant, which I abbreviated SPT here, would include the solubilized proteins. And that's what we're interested in using in the page process. You should also go check out this YouTube up here and just watch it to 1 minute and 47 seconds. This is also on the VLO sheet. Because page and Western blots are such a core molecular technique, we can see some different ways that scientists will use the techniques. So one way is to run something called just a page, which we talked about before, polyacrylamide gel electrophoresis. More often in this class, we're going to almost always get to see SDS Page. And so SDS is a strong detergent, and what that means is that this can denature all the proteins. So this is called a denaturing gel. So you should be able to use all those terms together. SDS stands for sodium dodecyl sulfate, and that's the strong detergent. So what SDS does is it disrupts the protein structure. Specifically, it causes proteins to unfold by coating them with a negative charge. And this is many, many, many negative charges, not just like a single little tacked on negative charge. And that means that the protein subunits will separate and stay separate from each other. You can see this in this drawing here. So basically this is the structure of SDS. You don't need to keep track of all the details there. By any stretch of the imagination, just want to show you a little bit about what it looks like. And this would be the hydrophobic tail, and then this would be the hydrophilic portion. And you can see the negative charge on SDS here. And basically, we see these two green squiggles here. These are each a polypeptide chain, and they're normally in a complex, and they're held together by these interactions. So S bonded to S, and do you guys remember what kind of a interaction that is? It should be a disulfide bond, and basically, as the process of SDS Page is set up, then the proteins will fall apart. You can see these little red dashes here, and if you want to see the details, look in your textbook or the other resources, and then you can see that there's little red dashes all around the polypeptide chain. So each of those is negative charge, and so that means that each of these polypeptide chains will repel one another. And again, this means that the two subunits will not be interacting when we look at the results of the Western blot. Likewise, though, even if we had a single subunit protein, like we see here, just a single polypeptide chain, it's also going to get coded with SDS, so we see those negative charges here. And so what I'd like you to do is to predict the migration of these different polypeptides A, B, and C in an SDS page. So draw it out as a lane here and a lane here, and then just draw some respective lines for migration, this being the top of the page gel and this being the bottom. So pause the video right now and take a moment to write that out. Okay, so basically we can see that the longest of the 3 polypeptide chains across here is B, so that one's going to be the most extreme position closest to the top. The shortest one is A, so I'd put that one down here, and then C looks like it's in the middle. And this would be predicting the migration in an SDS page. Now check out the this YouTube video, which again is on the VLO sheet. I jump around a little bit. You can certainly watch the whole thing, but you will hear extra details which may or may not be confusing to you. And basically just watch the first 35 seconds, skip ahead to 128, and then watch till the end. One thing I want to point out that I think will help you understand this process is that when we run the page gel, basically there's tons of proteins that are going to be separated across the whole gel. And so if we were to use a blue stain, which just stains all proteins, and looked at the gel at the very end of the page, so you actually add the stain at the end, but anyway, you're going to skip the transfer and Western blot steps, and basically this is what you would see. This is very typical. This is our molecular weight size standard, and then these are all the different conditions, and you see that there's lots and lots of different proteins. And just for your information, you don't need to memorize this. The stain that's used is called Kumasi blue. If you, instead of adding this blue stain, would do the transfer and then use a specific antibody and do the rest of the Western, then you would, for example, have an antibody that bound just to this one protein that goes across here, which I've sort of outlined in green. Then you would only visualize that one protein, and everything else here would be white.